Welcome, and thank you so much for joining us today for our second webinar of the year. We are going to be talking today about brain aging, Alzheimer's, and resilience, and we have two wonderful speakers, Dr. Miranda Orr and Dr. Randy Bateman, and we'll do some introductions in just a minute. But first, my name is Meg Smith. I'm the CEO of CureAlz, and I'm really pleased to be today's moderator. We are grateful that so many people are able to join us today, and we'll be watching this on a recording. So we will be recording this, of course. We know that each person who joins today or watches later will have their own reason for being interested in Alzheimer's, but that we are all united in wanting an end to the burden that this disease puts on so many people in our immediate circles and in our broader communities. Cure Alzheimer's Fund was founded about 22 years ago by families who had loved ones afflicted with this disease. And even though the disease had been known for about 100 years at that point, they were shocked that there were no better opportunities for treatment or cure than simply trying ibuprofen. They were determined to change that, and so they began Cure Alzheimer's Fund to accelerate the research that is necessary to do the discovery science and accelerate the therapeutics and treatments that can bring hope and better outcomes to the people with Alzheimer's today and in the future. We are intentionally supporting bold ideas and novel hypotheses, and all of our scientific decision-making is done by leaders in the field like our two guests today. Thanks to the support of our board, trustees, and certain other donors who choose to support our operations, 100% of all other general donations go directly to our research program. To date, we've committed over $270 million to scientific projects around the world, across more than 1,100 research grants, and we're really proud of all of the discoveries that these projects have enabled. Before we begin, again, this session is recorded. For other housekeeping notes, we are going to ask that you submit questions at any time during the Q&A feature, and we're going to reserve some time at the end for questions and answers. And I'm also going to thank Dr. Bateman again for joining us because he is a little bit under the weather, and so his voice is a little bit raspy, and we're going to try to give him some rest in between his answers. So with that, let's get going with today's topic. As most people will be aware, aging is the biggest risk factor for Alzheimer's disease, and for many people, the two seem to be really intertwined. But we also know from working with and knowing and loving wonderfully engaged and completely cognitively intact seniors that Alzheimer's is not an inevitable part of aging. So we want to understand, how is it that people stay cognitively healthy, and how can we offer that to everybody and not just the lucky few? To address this question, Cure Alzheimer's Fund launched the Brain Aging Consortium in 2025, bringing together leading researchers and labs around the world across genetics, neuroscience, and aging biology. Two of them are here today, and they are focused with their colleagues on three key questions. How does aging increase Alzheimer's risk and why? How could we detect Alzheimer's disease and see how it differs from aging as the process starts? And what separates healthy brain aging from disease? And how do we protect and preserve healthy brain aging? So to tell us more about their work and that of the Brain Aging Consortium, I am so glad to have Dr. Randy Bateman and Dr. Miranda Orr here today. Dr. Bateman is chair of the CureAlz Brain Aging Consortium and a leader in Alzheimer's prevention and biomarker research. He is the Charles F. and Joanne Knight Distinguished Professor of Neurology at Wash University of Medicine in St. Louis, Washington University, where he detects the dominantly inherited Alzheimer's network. His research focuses on understanding the biology of Alzheimer's disease and advancing new diagnostics and treatments, particularly through studies of individuals with inherited forms of this disease. And I'm eager to hear more from Dr. Bateman on the distinction between inherited and non-inherited Alzheimer's disease. Dr. Miranda Orr is also a member of the Brain Aging Consortium. Her work focuses on the biology of aging and its impact on brain health, and in particular the role of tau pathology in Alzheimer's disease. She is an associate professor of neurology at Washington University of Medicine in St. Louis, and her research on the biology of aging and tau-related neurodegeneration has explored the role of cellular processes that drive Alzheimer's disease and related dementias, and in particular cellular senescence. And I'm eager to hear from Dr. Orr how we define cellular senescence and how we're thinking about reversing or addressing it. We're really fortunate to have them here with us today to share their insights on brain aging and Alzheimer's. And welcome to both of you.
Thanks, Meg.
So, if I could start with you, Dr. Orr, can you tell us about why aging is an appropriate topic right now? After all, we've known about aging as long as we've known about humans. So, why is this an opportunity at this particular moment in time?
It's a great question, and I think there's a lot of reasons. One of which is because we actually know a lot about aging. It's not a passive process. The biology of aging field has spent a lot of time and effort really defining aging from a biological perspective. So over the past decades, we've gained data, and uh, the field in general has come up with hallmarks of aging. These are 13 fundamental processes. Well, depending on what paper you read, 9 to 12 to 13. And these processes are conserved across organisms. They are fundamental processes that not only co-occur in our cells and our tissues as we get older, but if they're disrupted, they can accelerate these age-associated declines. So you can have disease and dysfunction much earlier in life if these fundamental processes of aging are perturbed. And on the flip side, the field has now identified ways to intervene on these processes of aging. And if intervened upon appropriately, we can delay or prevent these age-associated processes from happening, meaning we get to enjoy longer, healthier life. So with all of these fundamental findings in biology, we can now actually study this across the lifespan.
Thank you. So, with these 9 to 12, depending on who you ask, processes, is that how people talk or defining aging clocks? What is an aging clock?
There's a lot of work on these clocks. So what clocks essentially mean in broad terms is a way of not just measuring chronological age, which is, based on the year in which you're born and the amount of time you are on the planet, but these clocks are more a prediction of how you are biologically aging. So an individual can have an older chronological age and a younger biological age, meaning that even though they've spent the same amount of time on the planet as somebody else, their biological aging might be slightly slower, meaning they're doing a little bit better job at avoiding what happens naturally as we just exist. So many stressors that we encounter, and all of these are very dependent on individual experiences. But things like whether or not we decide to exercise, our diet, I know that's a topic that a lot of people are interested in, our professions, our hobbies, all of these things influence our biological aging, but that doesn't necessarily change anything about your chronological age.
So, in your bio, I got to highlight your work in cellular senescence. Now, first of all, if you could define senescence for our audience, that would be great. But also, cellular senescence makes it sound like maybe our entire body doesn't age all at the same time or at the same rate. What do we know about that?
Cellular senescence is one of the hallmarks of aging. So it is a very complicated stress response. So when your cells are experiencing a variety of stressors, they have pathways that they activate to protect against the stress or resist the stress or even fix the stress. So if cells are undergoing a constant stress and they're unable to repair this stress, sometimes they die, and cells can die in a lot of different ways. Sometimes they may continue to divide, and that is how cells become cancerous. However, what cellular senescence is, it's not cells dying or proliferating. They completely change their cell state, their cell fate. They become something very different. And they have upregulated processes that make them want to go down the cell death pathway because of the damage, but they simultaneously upregulate these really strong pro-survival pathways. So these are very sick cells, but they stay alive. And as we get older, we encounter more and more stress, so your cells have more opportunity to enter senescence. And the only way these cells are cleared from the body is by the immune system. And so the cells do release signals to the immune cells to come and clear them. But if the immune system doesn't hear these signals, these messages that the senescent cells are releasing contribute to inflammation. And so now you have an environment around this sick cell that is very inflamed, and this can perpetuate a lot of other cells to die or become senescent or dysfunctional. And what happens is in every tissue that's been studied so far, we have identified, or the field has identified with aging, an accumulation of senescent cells. And these senescent cells are appearing near pathologies. They are co-occurring with diseases. And in laboratory studies, if these senescent cells are removed, selectively removed, it delays, sometimes even prevents some age-associated diseases and allows the laboratory model to live a longer, healthier life. And to your other question, is it true that some organs can age at a different rate than others? That's other work that has been discovered in the field, including by one of our consortium members, Tony Weiss-Correa, has shown that different organs can age biologically at different rates, which is very interesting biologically. And I also think it has a lot of implication on when we start thinking about therapeutically targeting aging.
Wow. So from your description, it sounds like there are at least a couple of opportunities with the senescence process. One is if we could keep the balance between cells kind of originating and going into planned cell death, keep that well balanced and avoid senescence. And the other is if we could maintain a healthy immune system that recognized senescent cells and cleared them out. Do we know what happens to the immune system as we age, or are there age-associated changes?
Definitely. There's a lot of age-associated changes that happen with the immune system, and they're very complicated. And I think an exciting area of research that is being more explored all the time is how the immune system is interacting with the brain. So this field of neuroimmunology is rapidly evolving. There's a lot of discoveries happening all of the time. And another important point with senescence, I think that it's worth stating, is that I don't think it's a good idea to prevent cells from entering senescence. So senescence is a way of preventing cancer. So if a damaged cell, if their options are death, senescence, or cancer, in the short term, we definitely don't want cancer. If they start accumulating as senescent cells, there are therapeutic opportunities to clear them, selectively clear them. And with that therapeutic approach, broadly termed as senolytics, that's the idea, is that we don't want to prevent cells from entering senescence, but clear them when their accumulation is causing inflammation.
Okay, thank you. So, Dr. Bateman, if I can turn to you, we just heard a lot from Dr. Orr about aging, but in your role as the leader of the Dominantly Inherited Alzheimer's Network, you work with folks who develop Alzheimer's disease much earlier than what we think of as being in aging. So can you talk a little bit about the distinction between aging and Alzheimer's disease?
Yes, I can talk about it, and I apologize ahead of time for my voice. So I have a hoarse voice for everybody. But that may be an example of the largest mismatch we have between organ aging and diseases of aging and the actual person's chronological age. So these family members carry a mutation that causes their Alzheimer's disease. Typically, it causes it in their 30s, 40s, or 50s. They have the symptoms of dementia, and they pass away. They die from that disease. Because they're so young when they get it, their bodies are completely young, healthy, and intact, but the mind is being ravaged by Alzheimer's disease. And so this gets back to what Dr. Orr was saying, that their different parts of the body, different organs, can age at different times. In fact, some of the mutations occur in genes that are called pre-senilin. Before people should be getting senile at a young age, these mutations in these genes cause senility before its time. And the clues to Alzheimer's disease, though, are held in families that carry these mutations, and how we compare that to the much more common late onset Alzheimer's disease that occurs in people past the age of 60. But when you look at the brains of these younger people with mutations, and when they have their dementia of Alzheimer's disease, they're quite similar and the same disease is the common form that we're all at risk for as we get older. And these younger people, they still get amyloid plaques, they get tau tangles, they get neurodegeneration, which is the loss of the synapses, the thinking cells of the brain's connections to each other, and the neurons themselves, the cells that do the thinking, those are lost as well, and characteristically in the same regions that occur in Alzheimer's disease of the late onset form. So here we have a way to disentangle the aging effect from the disease effect. You have young people who get Alzheimer's disease, and you have young people who don't get Alzheimer's disease. Similarly, we have older people who don't get Alzheimer's disease, but some older people who do. For those of us who see Alzheimer's patients or knows people with Alzheimer's, we appreciate that sometimes you can have a perfectly healthy body, you can have a strong heart, great bones, good eyesight, you can be vital physically, but your brain has been affected so much by the disease, it can be the reason that that person passes away and dies and lose their independence. Conversely, we have people who can live to old ages and have a ton of physical problems, but their minds be completely intact. And so I think this is a nice demonstration of how these two factors are really, in some ways, partly independent from each other. But if we can understand what it is about brain aging that leads to the risk of Alzheimer's disease, then that's a clue and a way that we might be able to prevent it for everybody. And more specifically, what I mean by that is if we understood why it is that people, as we get older, the risk of Alzheimer's goes up and up and up, it doubles every 5 years past the age of 60, we might be able to treat those reasons in a way that lowers everybody's risk of Alzheimer's disease, leading to pathways for prevention for the entire population.
So as the chair of the Brain Aging Consortium, and you've touched on this already, but what kind of comparative studies are you doing? What are you looking at, and in what kind of groups are you doing that to try to disentangle this as you describe?
So there's a variety of studies and investigators in our consortia. Dr. Orr is one, and I'm going to ask Dr. Orr to help me out here a little bit as we step through this. I'd like for this to be a little more of a fireside chat and dialogue where I get lots of help because I need it. But in general, we have 6 centers, 6 labs, principal investigators that are part of this brain aging consortium. And these centers, these labs, work on different parts of the problem. We have this beautiful spider web diagram where we're all connected in 6 or 7 different ways to each other. And what we're doing is we're taking each of our individual expertise and we're working with the 5 other groups to synergize and bring them all together in a way that answers the big questions that we just talked about. What is it about aging, brain aging in particular, that leads to the risk of Alzheimer's disease? Can we figure out what those mechanisms are? And if we can't figure out what those mechanisms are, can we think of ways to target or treat those brain aging mechanisms to help prevent the risk of Alzheimer's disease? I'll mention just a couple of labs, and then I'd like Dr. Orr to chip in and help me out with the others. We have Dr. Li Wei Tsai's lab is leading some work. Dr. Orr's lab is leading work. She'll describe her work. Dr. Henne Holstage in Europe has a centenarian study, people who make it past 100, and they remain remarkably cognitively intact. What's so special about them? My own group focuses on proteomics, looking at these clocks, the protein clocks of the brain, and what is it about the protein pathways that change with aging. We also bring some of the expertise in for these early onset cases where people get early onset Alzheimer's, and we contrast and compare that to the Dr. Holstage's cases where they live to be 100, and don't have Alzheimer's. So what is so dramatically different between those two groups? And then Dr. Andrew Yu's group has this in vitro system, which is it's a culture. You actually can grow not just cells in a dish, which I think most people know about, but you can actually turn them into neurons and then study how those neurons work with each other. And Dr. Orr mentioned Tony Wiscore's group, looking specifically at the generation of CSF. Miranda, do you want to comment maybe a little more, though, about how we synergize and how we work together and how we share things in a way to have a bigger impact?
Yeah, it's such a great group of incredibly talented scientists that are tackling Alzheimer's and aging from different directions, as Dr. Bateman just said. So some of the cohorts that we've brought together that I think are incredibly powerful are, like Dr. Bateman said, the Diane studies. So these individuals that are carrying genetic mutations, and we know when they will get Alzheimer's disease. And then we have on the other end of the lifespan, these very successful agers or the super agers from Henne Holstig's cohort. And so I'll talk about my project first and then how I'm intersecting with the different collaborators. In our lab, we have looked at brains from people that passed away very early in life. Age 16 is the youngest brain donor that we had, all the way to 116 was the oldest brain donor that we had. And so we've been looking at hippocampus, which is a part of the brain that is really important for learning and memory, and it's impacted in Alzheimer's disease. And so we looked at about 200 individual brains across the lifespan that either died when they were cognitively healthy, so they didn't have any functional impairments, or that they passed away with dementia. And we looked at about 100 proteins in neurons in these individuals' brains, and we asked the question, what is the pattern that happens with these proteins just across the lifespan? And then how do these proteins maybe change with Alzheimer's disease? And so now we have this reference data set. And one of the questions that we want to ask within the Brain Aging Consortium is, do individuals that are carrying these mutations that we know will predispose them to get Alzheimer's disease much earlier in life, do their neurons actually appear to be aging faster? So getting back to one of your clock questions, is their clock a little bit faster than a clock of an individual that doesn't have a mutation? And then on the other hand, with Henne's cohort, these individuals that lived 100 plus years old with normal cognition, is their clock set a bit slower? If we look at their neurons, do they appear, even though they passed away when they're 100 plus, do their neurons actually look like a neuron from somebody early life or midlife? So that's a very interesting question we're trying to understand. And part of that is what proteins or what are the machinery in these cells that may enable them to do that. So that's how I'm interacting with those two groups. And then interacting with Andrew Yu. So Andrew is able to take cells from individuals and turn them directly into neurons, and his method in doing so preserves their cognitive age. Or preserves their biological age. So something that we're really interested in knowing is whether or not those cells in a dish mimic the cells in the brain, and if so, at what age in the lifespan do those cells mimic? So that's a collaboration with Andrew's group. With Dr. Bateman's group, they're looking at the CSF from these same individuals. So we want to see if the proteins that are ending up in the CSF are also proteins that are changing in the brain, and if so, what cells are important for those changes. So trying to match our cohorts there. And then with the really incredible mechanistic study that Tony Wiscore is doing, actually looking at what proteins are being produced by choroid plexus across ages, trying to make a match there. And then Li Wei Sai's group, she is incredibly expert in multi-omics. And so one of the approaches that she's bringing to this group is lipidomics. So we've been talking about protein, and there's a lot of work on transcripts, so how genes and gene expression changes with age, but an underappreciated or an understudied macromolecule is the lipids. And so Li Wei's group has really been advancing that area of research. So we're going to match lipidomics, what's happening in proteomics. Again, we're trying to match brains from all across all of the different labs so we can have multimodal data across the lifespan to really get a better picture of what's happening.
So this is just a fun way for us to show how we all work together. This is the little spider web that we were talking about. And look at this, 22 planned interactions across the 6 investigator teams. And it's even color-coded. We have this analysis collaboration where we work on the data. We have data sharing, like the green lines, where we actually transfer data for the other group to use. And then we have sample sharing. So from each of our studies, each of our work, we're actually all working together, sharing samples and data and analysis, and we get together on a regular basis and talk about brain aging, which is our favorite topic. And then we get together at different times in person and have Miranda took us to a treat, the oldest restaurant in the city, because of course we're an aging group, so we have to go to the oldest place anywhere. So it's delightfully fun doing this kind of work. And I want to point out the 3 different dimensions here. So you have this tissue modality model dimension, right? Are we looking at people's brains? Are we looking at cell cultures? Are we looking at scans? And then you have this other dimension, which is the disease dimension. And this is how far advanced is it? Is it before the plaques form, or once plaques begin forming, or later when the tangles form and the neurons begin to degenerate? What stage of disease is that particular tissue or modality or model from? And then the neat one here, this thing that's really quite different, is this aging dimension. And you see that. And so now imagine you have someone who's moving along in disease and another person who's moving along in age, and another model which may give a different insight. And you can see using these 3 dimensions, it allows us to triangulate in and cover a lot of space for what's happening. And our job as scientists is to disentangle all this complexity and find out what's causing what and what can we do about it. And that kind of effort, that kind of big challenge, really requires this team consortia effort. It's not really plausible for a single group or a single investigator to tackle something like that on their own, but working together gives us a much better shot.
So it's such exciting work, because what you all are doing is looking at both brains with and without pathology at 3 different life stages. You're talking about people who are middle-aged, who either have early onset familial Alzheimer's or they don't. You're talking about people who are sort of in the middle to late part of aging, who either are healthy controls or have Alzheimer's disease. And you've got folks from the supercentenarian group who are cognitively incredibly young for their advanced ages. But some of them still have Alzheimer's pathology. So I'd love to talk for a minute about how your group is looking at one of the really tough questions in Alzheimer's. Scientists in this space have had a really hard time with the fact that we define this disease biologically by the presence of beta amyloid plaques, neurofibrillary tau tangles, and neurodegeneration. But we know from lots and lots of autopsied brains that there are people who die with some Alzheimer's pathology but don't seem to have any cognitive symptoms. They don't have the dementia that is the symptom that develops from Alzheimer's pathology. So can you talk about how you all are thinking about resilience and resistance in your projects?
Yes, so it's an incredible observation, and something that we are very focused on understanding biologically. So when we talk about resistance, what we're thinking about in those individuals are people that have aged that don't have pathology. Somehow they are resisting the presence of amyloid and tau in their brains. Some of these individuals maybe have amyloid, but they're resisting tau. Regardless, they are cognitively healthy. And so that is a subset of the individuals in the supercentenarian cohort. And then there's individuals that are resilient. So they didn't resist the pathology. They actually have the pathology, but they have maintained their cognitive function. So that's also incredibly interesting and impactful if we can understand what that is. So with the different modalities that each lab is studying, so by combining transcriptomics, proteomics, lipidomics, not only in individual brain cells from these cases, but also in biofluids, we're trying to figure out what are the molecular features, what are the molecular tools and tricks that these individuals are using to either resist or become resilient to pathology. And I think something that
Miranda, I have a question. Of the two, which would one rather have? Would one rather be resistant to getting the pathology in the first place? Or would you rather be resilient so that if you get the pathology, you can still function normally?
well, if we get to choose, I would prefer being resistant and then hoping that I convert to resiliency if I actually get pathology. And I mean, that is a very difficult question to ask in cross-sectional studies where we have brains at, individual time points. But I would imagine that those individuals also exist. They were resistant to pathology for much longer than predicted for their age. And then maybe they did become resistant once they did get pathology.
Yeah, so we do have these biomarkers where we can track the pathologies, right? And we know that some people can resist pathology, and there's different kinds of pathologies, isn't there? So there's the amyloid pathology, and then there's the tau pathology, and some people resist one or the other or both pathologies. And then, as you said, some people are resilient even if they get the pathology. They somehow are able to function normally, think normally. And there's this one exceptional case we reported on in Diane, which is the young onset family mutation carriers. That person lived 25 years past the time he should have gotten dementia of Alzheimer's disease. And what we found when we studied him in depth was that he had these plaques. The amyloid plaques had been there for many years, over a decade, but he was somehow resisting the tau pathology. The tau tangles were not coming up in his brains. And he stayed normal for 25 plus years past the time he should have had dementia. And so that's a bit of a miraculous case. But there are also other people who are either resisting the pathology and resilient. I'm with you, by the way. I hadn't thought of the answer, but I'd love to have both resistance and resilience. It's a good thing to ask for. I think it's a rare thing. It would be hard to get both. But I wonder what our audience would want for it. Would you want to resist the pathology, the resilience, or ideally both? I agree. But it changes, that distinction changes, doesn't it? How we think about our research and how we think about how do we go in and prevent people from getting Alzheimer's and dementia. Because one prevention strategy is, do we resist the pathology to begin with? Do we not let it get started? Or is it okay if maybe if that's too hard or not possible, is it okay to think about in parallel to that or instead of that even, could we be resilient to it if we understood what it was that made these people so exceptional that they could live normal lives even with the pathology of Alzheimer's in their brain? Then maybe that would be a thing that we could develop and offer to other people to be resilient. Because at the end of the day, most of my patients and families, they tend to care about, they get worked up about things like plaques and tangles when we talk about it. But in truth, what they really care about is they care about their independence. They care about their thinking, their memory, their function. That's the ultimate goal. So I tend to agree that any way we get there is a good way to do it.
So Randy, I want to keep talking about that topic, but I also want to back up just for a second because you're one of the world's pioneers in blood tests for Alzheimer's disease, and you use the word biomarker. And I think not everybody in the audience is probably aware of what a biomarker is and what it is, how powerful it is that we now have blood-based biomarkers for Alzheimer's disease. Can you touch on that a bit?
Sure. So a biomarker just means a marker of biology. And at its most basic form, our blood pressure, our heart rate, those are biomarkers, markers of our function. What I was referring to, though, was more about the biomarkers, the biological signatures that we can measure, and things I mentioned, the PET scans where we can measure the plaques and tangles of Alzheimer's disease with a scan. That's a biomarker. We have MRIs and CAT scans. Those are biomarkers. We've had cerebrospinal fluid biomarkers for several decades. That requires a spinal tap, and not everybody's enthusiastic about that. So the field has developed these blood-based biomarkers where you can measure the proteins that make up these plaques and tangles in the brain. You can actually measure that in drops of blood that you just do from a single blood draw, and you can analyze the amyloid beta that makes up the plaques and the tau forms that make up tangles or reactions to plaques. And we can know something about the Alzheimer's disease pathology by using these blood-based biomarkers. And those have advanced to the point now where they're used in clinics. Doctors order them all the time to help diagnose our patients, help determine, does this person have plaques? Well, one day soon, we think we'll have tangle markers available to doctors. And then if we have those, then we can understand, does this person have plaques? Do they have tangles? If they do, does that support a diagnosis of Alzheimer's, or does it rule it out? And where this is ultimately going one day is if we have treatments that can be effective in prevention, because these plaques build up 10 to 20 years before the dementia begins, the symptoms begin, we might be able to actually identify people who are at risk of getting Alzheimer's, treat them before they get dementia, and prevent them from getting the dementia. Currently, that's our best game plan for how to prevent the disease. And it won't be that long. In just a few years, major trials will be reading out. We reported one of the first trials showing that when you remove plaques in people, that they are protected in the mutation carriers, these rare families, they're protected from their future onset of dementia. And so if that works for the general population, that could be a strategy to help lower down or prevent the numbers of people who are at risk for Alzheimer's disease dementia.
So the biomarkers are really an opportunity for us to know more about the future, we think. I mean, it tells us a lot about the now and hopefully then about the future. How is that different from what you can learn from a genetic test?
Well, it's different in two ways. The main way is a genetic test actually is a biomarker also. It tells us what the person's genes, what they started with as they were conceived in their whole life. So what we're born with is the same set of genes we have when we die, basically. And it comes from our parents, of course. And so you can test for what those genes say, and the genes could tell you about risk of Alzheimer's disease. The most common genetic form that is tested for is a protein called apolipoprotein E, and we call it ApoE for short. It's a genetic test that tells us about the risk, and there are three forms of it. ApoE3 is the most common form, and about 2/3 to 3/4 of people have that. And then there's these rarer forms, ApoE4 and 2, and 4 relative to E increases your risk of Alzheimer's by 3 to 5-fold for each copy you get. And ApoE2 cuts it in half. So we call E2 the protective form, E4 the risk form, and E3 the normal form. So we can test for that, but it just tells us about risk. It doesn't tell us about that person's state at the moment. So is it when you were born or as a child or a young adult, we could test for ApoE. We could tell you your risk, but it doesn't tell us if you're going to get Alzheimer's or when you're going to get it. And that's where the other biomarkers turn out to be more informative, because they tell us at any given age, if you test everybody when they're 20, almost nobody would have any risk, any Alzheimer's pathology in their brain. But if we test people when they're 60 or 70, then 5, 10, 20% would have the changes of Alzheimer's disease in their brain. And so that one is dependent on what that individual, what's happening in the brain of that individual. And that's what distinguishes them. One is a detector of a state, and the other is a detector of the risk. I'll comment that Miranda's own work, the power of biomarkers is so important in order for us to not just detect the disease, but but to track the disease, to understand the disease in living people, that, for example, the senescence that Miranda's team works on is developing new biomarkers for senescence. And that's exciting to me because once we have a way to track senescence in the brain in living people, now you have neat ways of thinking about, Miranda, you tell them. You're leading the work.
Yeah, so and Randy was just talking about ApoE, and so that's such a nice tie-in with senescence because there's work from others in the field that have shown that people, well, in mouse models at least, and there are some human studies that support this as well, that the ApoE4 allele or variant that increases the risk of getting Alzheimer's disease also increases the likelihood that immune cells will become senescent. And because immune cells are so important at keeping senescent cells out of our solid organs as we get older, if our immune system becomes senescent, that's really bad because not only is our immune system not functioning properly, but it's also having a huge impact on our organs. And so some of the work we're trying to do is, as Dr. Bateman said, is identify new biomarkers of these hallmarks of aging. Once we have identified them within cells in brain tissue, then can we determine if any of these markers are showing up in fluid, so then we can track it across the lifespan. Ultimately, what we would like is a blood test for knowing how your aging trajectory is looking. Like, is your blood telling us that your brain health looks really good and you're doing great? Or maybe you're not doing so good and we should consider some lifestyle changes or maybe even pharmacological interventions.
So look at this. So here's chronological age. This is what Dr. Orr described. How many years has one been on the planet for? And here's the model predicted age. And look at the line and look how all the dots, these are actual. So this is what the model says the person's age is. And this is what the chronological age is. Now imagine if you're one of these dots and you're right on the line. Well, then you're average. The model predicted you'd be 67 and it turns out you're actually 67. Then great. Well, how useful is that? We already knew you were 67. You didn't need a model to tell you that. By the way, this model measures proteins in the cerebrospinal fluid, brain proteins. So just by measuring your brain proteins, we can tell you about how old you are. Isn't that crazy? Like, have you ever been to the fairs where they try to guess your age? If you'd be willing to do a spinal tap and we measured proteomics, we could guess your age with some accuracy. But the interesting part here is that sometimes this person here is 67, but they have the brain proteome of a 57-year-old. And that's exactly what Miranda was describing. So we think the brain proteins in that person, their brain is younger from that protein expression level. And the question is, what makes them younger? What makes them behave that way? And so if you have this for these different processes, like senescence and other things, does that then enable us, scientists, and ultimately one day, hopefully in the clinic, to say, hey, your brain is doing really great. It's a really young brain, and keep doing what you're doing. Or worse would be, hey, you have the brain of an 85-year-old, but you're actually only 65, and that's a problem. And we need you to change what you're doing, and to, whether it's a treatment or whether it's a lifestyle change, exercise or certain diets or supplements or something, like, can we figure out for the individual what they need along the way? And I think that's the power of these biomarkers coming full circle, right? That if we can track what's actually going on in our bodies and our minds and our brains, it gives us an opportunity to do something about it. But if you don't know, then all we can do is offer general advice: exercise, eat well, stay socially and cognitively stimulated. I hope everybody's heard those messages. They're important messages.
Well, and I think they can't be repeated often enough. And this is a great time to segue into Q&A. We could keep talking science all afternoon. We didn't even get to epigenetics, so I'm sorry for that. But we do have some great questions. So one of the questions that's been submitted is on what is the role of cortisol and what are we supposed to know about it? You know, you talk about lifestyle choices. what does cortisol relate to that?
Is it okay if I talk science again? Like some
Yeah,
cool
absolutely.
So I will leave the clinical answer for Dr. Bateman, but I'm going to take this opportunity to talk about senescence again because I think it's such an important and fascinating topic. But there's been studies now. So cortisol is a stress hormone and it increases when an individual is stressed. There's been some very elegant studies that show that not only does stress, psychological stress, increase senescent cells in the brain, but the type of stress one experiences can impact the different types of cells that become senescent and the molecular tools that they use to enter senescence. So it's turning out to be incredibly nuanced, but the end result is all the same. These are very stressed cells. They can't figure out how to repair it. They become dysfunctional, and now they need to be cleared, where we need a healthy immune system to do that, which stress can also impair the immune system. So I would say that it's very nuanced. Cortisol in general is very nuanced, and how individuals handle the stress also seems to be very nuanced and an incredibly active area of research right now.
I agree wholeheartedly.
Okay. So if you could only give somebody maybe 3 pieces of advice about how to have a younger and healthier brain, where would you focus? What should each person be focusing on? Because everybody's getting inundated all over the internet about the ways they're going to live forever. So what's real and what's hype?
Sleep, eat, exercise. Those would be my 3.
Exercise, sleep, eat.
Okay.
But
So when we say eat,
can I make a point though?
yeah.
I used to write prescriptions for my patients back when we had prescription pads to exercise and to exercise 3 times a week, 30 minutes at a time, aerobic exercise. So I believe that the evidence for regular aerobic exercise, at least starting in midlife. So if you're over the age of 30, and I recommend it at all ages, but I highly recommend that for preventing, the effect size is quite large. People who do that on a regular basis has half the risk of dementia and Alzheimer's disease, half. And so it's one of the biggest things I think an individual can do. If you're really worried about getting dementia, I believe it's aerobic exercise. That means getting your heart rate up and sweating and breathing hard. It doesn't mean walking is great for you, but there's little evidence that kind of exercise or muscle building exercise is a strong protective factor against dementia. There's lots of biology that happens when we work out hard. Growth factors go to the brain sprouts new connections, all kinds of things happen. So I would almost list that as 1, 2, and 3, and then eating. Sleep, actually, there's a ton of work being done in sleep, as Dr. Orr said, and that actually is turning out to potentially be a big risk factor as well. I don't think we know as much, though, that intervening in sleep will decrease your risk of Alzheimer's disease, as much as people who get bad sleep are certainly at risk. If you have interrupted sleep, if you have sleep apnea, if you have these sleep problems, those people have increased risk of Alzheimer's, but it's not universal. There's some people who sleep very few hours naturally and normally, and they don't seem to have much of an increased risk of Alzheimer's. So there's something about interrupted or bad quality sleep. And there are actually studies going on right now, prevention studies, people can sign up for, and you can take good, like natural inducing sleeping pills to see if it can prevent Alzheimer's, plaques, or dementia. And so those studies are underway.
We recently launched a Circadian Rhythms Consortium that's looking at exactly these questions. And there is, it's a relatively small study, but there was a study last year that showed that treating insomnia returned individuals' risk of dementia to the same risk as people who didn't have insomnia in the first place. So that's a positive, obviously, because we want to believe that these are modifiable risk factors. So, Dr. Bateman, you talked about the blood tests. now that always leads to, and we've just gotten a question about it. Okay, if somebody does find out that they have a positive result in the blood test, meaning that they are, there is a likelihood of Alzheimer's disease, what can they do? What treatments are available now?
Oh, they might be eligible for attempting to remove the plaques out of the brain. So our current blood tests are largely detect the amyloid plaques in the brain. And so if someone has symptoms of forgetfulness or memory problems or mild dementia, they could go to a specialty clinic, like a neurology clinic, where they have these treatments. We've instituted these treatments, for example, in our clinic. And if a person has amyloid plaques and they have the symptoms of Alzheimer's disease and they don't have contraindications or reasons not to be treated, it's possible we have these in treatments now, which are infusions of antibodies. They go into the bloodstream and into the brain, and they grab onto these amyloid plaques. The immune system removes the amyloid plaques. And people who have that done are relatively protected compared to people who don't. And so the average number is about 30% and less decline than people who don't remove the plaques. That said, we have some people who are at extremely early stages, and they can have even larger effect sizes, 40 to 50% protection against the decline of dementia. And these are our first real, what we call disease-modifying treatments, treatments that change the disease itself, in this case by removing plaques and benefiting. We recently heard good news that was announced that there might be another class that's coming that targets tau, the protein that makes up these tangles in the brain. And so I think we're approaching an era where it used to be, I was taught in medical school and residency, that you can only diagnose Alzheimer's disease after someone passes away, and there's absolutely nothing you can do about it. And then we've gone from that to a day where we can diagnose people with a simple blood test and a clinic visit and remove one of the pathologies of Alzheimer's disease out of the brain, and maybe soon both of the pathologies. And so what this means to me is that we're now in an era where we might be able to do something about the disease that we could never do before and change the course for our individual patients in the clinic. And to me, that's one of the most exciting and profound things. This is the start of treatments and preventions, I believe, for Alzheimer's, but it's not the end. And there's much more to be done. We have to be able to deal with the immune system. We have to understand how sleep, how aging itself, senescence, and brain aging, we have to understand this in order to cover all the things that lead to Alzheimer's onset and progression. If we're ever going to really, the C word in medicine, the cure word, if we're ever going to cure Alzheimer's, I think that's what it's going to take.
Mick, can I say something on?
Please jump in.
Yeah, Randy inspired me to say something. So, as he was talking about, the amyloid, the tau, and the neurodegeneration. So the Alzheimer's field, from my perspective, when they coalesced around this fundamental framework, ATN, amyloid, tau, neurodegeneration, it really focused the field on what they were going to spend their time investigating, what they wanted to find biomarkers for, whether or not that was in the blood or brain imaging, and then eventually treatments. And that's, what Dr. Bateman was saying is we're there now. And so an idea that we're tossing around in this brain aging consortium is whether or not we can come up with a similar framework for brain aging, where those of us that are studying it can really coalesce around foundational aspects of brain aging and focus our attention and do for brain aging exactly what the Alzheimer's field has done for dementia. And so I think that could have a fundamental shift, but it's really hard. So right now in our meetings, we're talking about what this framework might be. We're working on a perspective piece and, presenting this to the field and getting opinions. So if there's anybody on the call that has some ideas of what they think these fundamental frameworks of brain aging, please let us know. Because I think it would be really important and would catapult the field in a major way.
Hear, hear, it would be transformational. So I completely agree with Dr. Orr on that. Yeah.
Well, getting to where there is hope. I mean, Dr. Bateman, you've told us now about drugs that can actually remove plaque from people who have early memory concerns. How close are we to having blood tests for people who don't have memory concerns yet, so that maybe we can intervene before there's any change in function? And Dr. Orr, you spoke about how there are people who are resilient. What's our best hypothesis for what allows people to be resilient, and how could we possibly replicate that for everybody?
I can answer mine quickly. We already have blood tests that can be used that can identify people who are at risk of Alzheimer's disease. And they're the same tests we use to identify people who have symptoms, whether the plaques are there. As I said, the plaques are there 10 to 20 years before the symptoms begin. So the tests are there. We don't use them unless people have symptoms. And people may be saying, well, why wouldn't you use them? The reason we don't use them in the clinic, we use them for research all the time. We use them for our trials. We don't use them in the clinic because right now the medical system, the guidance is, well, if you come in and you don't have any symptoms, then why are we checking you for Alzheimer's? Well, we could check you to see if you have plaques, but even if you did, what would we do about it? And that's the issue. We wouldn't know what to do because the trials for the main trials for prevention haven't read out yet. So we don't know if removing those plaques is helpful or does nothing or is harmful at that stage of disease. Now, some of us, maybe many of us, think it's likely to be helpful, but we don't know that. And in medicine, we have a long history of doing things because we think they'll help, and it actually ends up hurting. And so for that reason, in our clinics, we don't recommend that people, unless you have any symptoms of memory or something, we don't recommend people get the test done because it might be positive. And then what are you going to do with that? Are you going to treat someone without knowing whether it's beneficial or not? And in today's medical system, the answer is no. We would not treat people without knowing whether it's beneficial or not.
And
So, Miranda, before you jump in, sorry, I asked you a question. I really want to hear the answer, but it's interesting then that there isn't an opportunity for that scared straight moment to go in and get that test that says, you know what, your amyloid is going up. You should be making all the best lifestyle choices you can because we're seeing this in your brain. Um, it's too bad that that's not an option at the moment.
Yeah, it's not, but part of this, look, I have mixed feelings about this myself personally. The professional side of me says it's not a good idea to go screen a bunch of people who don't have symptoms to scare them straight so that they do the things they should be doing anyway, right? We should be doing aerobic exercise anyway, eating right anyway. But on the other hand, I can see reasons why individuals might want to know that and why that might change their behavior and might help them out. It's just the standard of the medical system, right? So, so I can see both sides of it, but I'm just trying to describe what is the standard medical recommendations. It's interesting. We do tests for other things that we don't have treatments for, don't we? And there are plenty of incurable diseases or treatments or things for which we don't have treatments for. We do those tests. But I think medicine has moved to a stage, especially with something as broad and as big as Alzheimer's, to where it's not acceptable right now to do that. And all the guidance clearly says don't test people unless they have symptoms for right now. Now, I personally think it's likely to change in just a few short years. And I think it won't be long before we are thinking about testing people before they have symptoms.
All right, thank you. And Dr. Orr, I would love to hear from you.
the resiliency question. So
Yes, please.
I know we're at time, so I will make this really short. We don't know all of the answers for resiliency right now, but it seems that what are keeping brains resilient are cells that we don't often think about in the brain, which is immune cells, as well as vasculature. So the people that seem resistant to amyloid pathology still have really good vascular health and little immune cell infiltration. So I think understanding neuroimmunology and how the vasculature is contributing with brain aging and Alzheimer's disease are two really important areas that continue to deserve attention.
Well, Dr. Orr, Dr. Bateman, thank you. We are at time, and I am so appreciative of the work that you are doing and the time you spent with us today. There is so much hype in this space. There is so much misinformation in this space. And it's really powerful to hear from people who are actually using phenomenal science to get to real answers. So we are incredibly grateful. This was recorded. We're happy to share it with anybody, and we encourage people to pass it along. And we'll look forward to hearing more results from the consortium as they develop. Thanks so much.
Thanks, Meg.
Thank you.